Welcome to MVIF Conversations, the podcast.
So welcome to a new episode of MVIF Conversations, the podcast. Today we are very happy to have with us three very inspiring voices from the ISALA project. Sarah Ahannach, Sandra Condori, and Josiane Kenfack. Be welcome. So the ISALA project is a pioneering citizen science initiative at the University of Antwerp, Belgium, and aimed at better understanding the female microbiome. And it has become a leading example of how citizen science can drive microbiome research forward while creating meaningful societal impact. We have heard about ISALA project and its sisterhoods in former MVIF events. So if after this podcast you want to learn more about what they are doing, don't forget to check our YouTube channel where you can find other sessions where they were presenting their work. Here in this episode, we are going to explore the project's ongoing work, the recent advancements, and the scientific insights emerging from this larger-scale community participation. We also reflect on the human side of the citizen science, from personal experiences, challenges, lessons learned, and the added value of engaging the public in the microbiome research. So I am Anna Cuscó, and I'm delighted to open this conversation. Before we dive deeper, I will ask you, Sarah, Sandra, and Josiane, to briefly present your roles in the ISALA project and share with us what drew you to this initiative. Maybe we can start with you, Sarah.
Thank you, Anna. Hello, everyone. My name is Sarah Ahannach, and ISALA is soon, in a few days, going to celebrate its 6th birthday. We always try to celebrate with cake. So I reflect back on the ISALA project as a project that was established together in the lab of Professor Sarah Lebeer. So together we co-founded the ISALA project. And then during my PhD, I worked on setting it up and studying the first mapping of the vaginal microbiome in Belgium and setting it up. And now, over a few years, I've grown into more of a ISALA program manager role. So I'm excited to see the team grow both nationally and internationally.
Nice. Thank you. Happy birthday to the ISALA project. Maybe now you can continue, Sandra. Let us know.
Yes. Hello, everyone. I'm Sandra Condori, and I'm a postdoctoral researcher at the laboratory of Professor Sarah Lebeer. And with me, the ISALA team, I am the project manager of the ISALA Sisterhood. And I'm also leading the Laura project in Peru.
Thank you. Thank you for the introduction. What about you, Josiane?
Hello, everyone. Thank you very much. So I'm Josiane Kenfack. I'm a PhD candidate working on the vaginal microbiome. Actually, I'm the co-coordinator of the ISALA Sisterhood in Cameroon named Leke Project. So my deep motivation on this project is to improve women health research and particularly underrepresented groups like African women. Thank you.
Thank you. Thank you so much for the very nice introductions. I think before moving a little bit deeper, we should start by the beginning. So for the listeners who might not be familiar with the ISALA project, I think it's a good point now to maybe, if you could start telling us a little bit about how the ISALA project got started and which were the main goals about it. Maybe Sarah?
Yeah, definitely. So a lot of research has been done on the vaginal microbiome internationally by some very nice groups. And our lab, I think specifically, was looking for better understanding the role of lactobacilli in the vaginal environment. This was also within the ERC starting grant of Professor Sarah Lebeer. And we felt that a lot of the data out there was always studied in the context of a university hospital. So a lot of pathogens, disease, and conditions on vaginal health were studied, and then there was a control group. These were the same women, different women, but that also went to the gynecologist in the university hospital. And they seem to have a lower amount of lactobacilli. And we were a little bit getting worried, thinking that maybe maybe they're less lactobacilli, maybe especially and same as in Belgium and in other countries. So we were mostly curious to know what does a healthy and optimal, or just a general vaginal microbiome of an average person look like. And this was something that we were a little bit missing, because we believe that when you start from knowing what is, let's say, I don't want to say too much normal, optimal, and healthy, because but just knowing what is the general, let's say, the same microbiome, then we can better understand how you maybe move into a disease state or come out of it. And for us, it felt like more of the foundation. And that were the first goals of ISALA, is to map the vaginal and female microbiome. We started in Belgium. And then next also to understand the environmental factors and the influencing lifestyle factors, life course factors on the vaginal microbiome. And then the third scientific objective here was also to build a biobank of live vaginal lactobacilli that could serve as potential promising probiotics and live biotherapeutics. So we started with these three goals as it comes from a scientific perspective, but immediately from the beginning, we also had societal goals. So we really immediately focused on breaking the taboo on intimate health, on empowering women to understand their bodies, and also on really promoting interdisciplinary science. So not just us microbiologists and microbiome scientists, but expanding to sociologists and clinicians, communication experts, and different stakeholders. Yeah.
Okay, yeah, super interesting. Also, like something that maybe started like more locally, it happened to be a need that somehow extended abroad. So, we will touch on all these things that you have said across the episode, because it's very interesting. But okay, what started locally, now this is not local anymore. Like, it's a very international thing. So Sandra, maybe that you are responsible project manager for these international projects. Could you tell us more about how these sister projects emerged? So how did ISALA expand beyond its original context? And what does this growing international network look like today?
Yes. Well, at that time, I think it was 2020, I joined the lab, I think a year before. So I was witnessing all the work that was being done for ISALA, all the preparations. And I was a newbie in the lab, but I really got excited about the topic, the approach they were following, the almost 6,000 women that registered. So I said, oh, okay, this is huge. This can be done in my country. And I checked literature. There was nothing about it in Peru. And I said, okay, no, we need to start to do this. So I applied for a small funding. We got funding for Laura. And also the big idea, let's say, started with talking with colleagues. I had a colleague in Switzerland, and she also got excited. Wow, this is what ISALA is doing. It's so cool and great, and with such an impact. And and then we decided, okay, let's start the Marie Sister Project in Switzerland with other colleagues also. I'm referring to Monica Tilla and Ethel Mendocilla. They really put a lot of effort to start Marie, because it started as an idea, but then you need to look for a host lab and all these things. Now everything is assembled, and then there is the host lab and everything. But it started like that, like dreaming practically. What if we do this in this country? What if, we extend this? And also at the same time, after Marie, Josiane was also following the team, the work, and we said, oh, and why not in Cameroon? Why not to start a project like this? So we applied for funding. We were lucky, and we got the funding to start Leke, and eventually, we received contact with other researchers around the world. And Sarah also, I remember our conversations about dreaming. Dreaming about the logos, the underwear logos. In the string. That would be really cool. And of course, Sarah Lever totally supported our dream of studying the vaginal microbiome globally.
Yeah, that's amazing. I mean, I always love listening about this story and how these things escalated so not easily, but because I guess it was not easy, but like so naturally, like there was really a need there that no one was covering. And it's very nice to see how this is spreading globally. So yeah, okay. Now let's talk about how to adapt these things to different cultures, right? Like how did you adapt the citizen science project across the different cultures? I think maybe we can start with you, Josiane. So which parts must remain consistent with doing this project and which parts need to be locally adapted? So for example, in the Leke project in Cameroon, how it was to setting the project up in Cameroon, what was different, what you needed to adapt?
It was really amazing to begin this project in Cameroon because it was the first time to use the citizen approach on human health in Cameroon. And at the beginning, we didn't know how human will react, but finally everything went well. And also we have at the beginning the same idea to know how the vaginal microbiome of Cameroonian women look like, but also we also add some topics related to Cameroon. For example, HIV. You know that HIV is still a public health problem in Africa and Cameroon. So for Cameroon, we work with healthy women and also we work with pregnant women, HIV positive and HIV negative at the hospital. And also the communication was different in Cameroon, and also the communication were added for each part. For example, in the urban area, we have to communicate, we have to have in-depth communication with women. So we didn't really involve women in social media, Instagram, like ISALA. We have to enroll women in women association group. We have to enroll women like in the circle of influence. Like I have my friend, I present the project to my friend will present to a friend. It was like that. And this is also because this is kind of like communication in Cameroon. And also, as I said, we have to, for each woman participating in the project, we have time to communicate with them, to explain the project, and also to raise awareness about vaginal health. But in the rural area, the communication was different because in the rural area, people live mostly in community. And first thing that we have to do is to have a discussion with a man, because in the rural area, it's important to have the men authorization before engaging communication with women. And also, one thing that we also want to do was to involve men in women health management. So we have this conversation with men firstly, and it was really good, and they were really involved, and they was really like to help their women. Then after that, we have this general conversation with women under the tree that is also a representation of our culture. So in general, that's how we managing the project in Cameroon.
Okay, yeah, it's interesting. Then even within the same country, you need to really adapt depending on the part that you're targeting, right? So, yeah, I mean, I don't know, were these challenges different from the ones that you found in Peru or Belgium? I don't know if you want to tell us something.
Yes. For example, if I compare with Belgium, what we did in Peru was to work with field workers, because in ISALA, for example, they deliver the kits via the postal service. And yeah, in Peru, this system doesn't work. It exists, but it doesn't work at all. So we had to work with field workers, and actually to motivate women, it was done in the same way with the word of mouth, communicating between groups of women. We also use social media, but not too much. Let's say we mostly focus on personal communication, let's say. And yes, and so this was the way it was done. Then another difference, I don't know if in Belgium they face this, because it was online mainly, since the field workers had to go and talk about the project in the community. In some occasions, they saw that the role of the mothers more than the husbands, or like in Cameroon, they sometimes were preventing some participants to donate samples because they were afraid at the beginning. Yeah, in some cases, yeah, there was a deeper conversation and they could understand, but in some cases, yeah, they just refused for their daughters, but they were adults, the daughters. But yeah, it's the influence sometimes of the mother. Yeah, but it's worse cases.
It makes sense.
I think also in Belgium, in the beginning, we were looking for 200 participants, and at that time, people were saying like women are never gonna donate a vaginal swab. They are. This is something that probably women don't want to do, and that's why you put a lot of time and effort into your story. And we were surprised that within 10 days we had 6,000 registrations, so we had to stop the website and so on. But when we were analyzing the results, a question also popped up, and who does this represent? Who are we telling a story about when you're writing your paper and so on, and when you're doing the communication? And then we started to, together with their advisory board, set up this demographic profile of the participant. And there we saw in the first stage, because citizen science is not a population study, so it is self-biased in the people that enroll themselves. We saw that these were women that were higher educated than average, had a higher socioeconomic status, were mainly white. We did have 10% of diversity and a few percentage also of women living in poverty, but this was smaller. So within our group, we had to focus on how can we, in our future enrollments, include women from different layers of society to have a more representative view. And there we worked together with sociologists, having focus groups and conversations with migrant groups, with women living in poverty, to adapt our communication style and some of our strategies, which has helped us now in the coming years to be more inclusive. And we're still not there, but I think you keep reinventing and learning from yourself. So that was, I think, one of a struggle that all of us will have to face. Everyone doing a study is a representation and balancing homogeneity and heterogeneity as well in your data. And then, as Sandra and Josiane were saying, also for expanding to the sisterhood, and this is Sandra can speak about the most, is it takes also a lot of administrative effort to include international partners. It's not as easy. Every country has their own system, their own funding system, their own administrative system, and we didn't have the funding to do it. So a lot has and up until now, I think, is still being driven by passion and volunteering. So you want to add to that?
Okay,
Yes, indeed. Yeah, would be nice to have funding to start that all sister projects start sampling. But yeah, some projects, for example, started with the online campaign, the science communication, raising awareness. But yeah, it is actually a lot of work and effort.
Yeah, we need much more funding. In fact, in women's health in general, I think there should be something to be solved, like from the public side, right? But let's see. Okay, so talking about these differences also among the different countries and everything. So specifically for vaginal samples, how do you engage women and encourage them to participate in this study that involves intimate sampling? So especially that's given that it's a taboo topic, how did you manage that? If there is something else to that. We got a little bit into it in the previous question, but if you have any other specifics.
I think from at the beginning of ISALA, it was a lot of testing our communication. So within the team and also with our environment. But we try to find the sweet spot of breaking taboo, but not going too over the top that it would halt particular groups, because sometimes it can get too much or too visual, or sometimes considered vulgar, because you can go different ways. And this was something that at the beginning of ISALA was done together with a communication bureau that was hired to help us start up in the beginning. And now we're developing also a bit on feeling, and by doing a lot of outreach activities, you get a feeling of how groups react. But we try to find a balance. And while also now studying and doing this focus group, you learn also how more and more. But in general, what I think is what I advise people would always to have in your advisory board also citizens or ambassadors from particular groups in society that can also give, because every group looks different at particular use of wording. So that was, I think, necessary for Belgium. But Sandra and Josiane can tell you more about how it was perceived in their countries.
Yes. Yeah, maybe for Laura, the way we approach or how we motivated women to participate was to raising awareness about good bacteria, because what it's very common in Peru is, okay, use antibacterial soap, you have to be clean and wash and wash. But yeah, by bringing this knowledge on, hey, you have good bacteria that keeps your health, and you have it in your vagina, and explaining this for them. I remember during the information sessions, it was like, oh, wow, really? Do we have good bacteria? And how do we keep them? How do we care these bacteria? There were some questions about it. And also, the taboo topic was all, but the taboo, yeah, it's still a taboo because most of them kind of didn't want to talk about it. But when we talk about what they feel when they go to the gynecologist, and that we were saying, hey, we are going to investigate this to gain knowledge, to know alternatives, for example, to the use of antibiotics, probiotic bacteria could do it, and explaining all this, and they say, yeah, it's good that we can contribute, because they felt also that when they go to the gynecologist, they don't get listened, they don't get alternatives, and they feel like no one is doing anything. Then if you will do something that will help me or my daughter in the future, even if I'm not alive, then I will do it. Yeah, I remember these conversations, yes.
Okay.
Yes. So for us in Cameroon, it was also like the same, the discussion with women about improve women's health, and also, as I said in the beginning, the thing that we do in Cameroon is that we have a deep conversation with each woman. I can say that with each woman we have like 30 minutes, maybe more 30 minutes, and during this conversation we try first to raise awareness about vaginal health, and also to make women aware that the vagina present in their the bacteria present in their vagina can play a great role in their health. Those bacteria can either improve her hair, or also those bacteria can sometimes allow make them to be allow her to have like disease like HIV, SCI. So we first make women aware on that, and also we also try to explain to women that actually in the research we don't have knowledge. For example, concerning Central Africa in general, where Cameroon is, based off my research, we don't have any information on how the vaginal microbiome of women is this in this place. And we explain to women that it's really important to have this knowledge, because if you don't know how the vaginal microbiome is, there are many women health issues that we cannot tackle, and women will feel concerned by that. And it was easier for them. And also we explained that we don't want to consider them only like a person who gave samples. We wanted to be involved in all the process. And to be involved, the first thing that we need for them is that they have to take their samples. You know that in research, sampling process is really important. If you ask for a woman to give you vaginal swab, and if, for example, they give you skin swab, you have a wrong result, and we say to the woman that you are at the center of the research, and we want you to take this responsibility to take your samples. And with this kind of communication, the women will really feel involved, and it was not difficult for when we say all these things, it was not difficult for her to take the sample. So we didn't need to convince her again. So I can say that all this turned around the kind of communication we have with the participants.
Okay, yeah, that makes sense. Like plenty of communication, giving them plenty of information, and making them part of the project, like active part of the project through all the projects. So yeah, that sounds very reasonable. Okay, so we have already touched a little bit about the common myths and misconceptions, like using these antibacterial soaps that you were telling us in Peru, Sandra. But are there any other like myths and misconceptions that you have encountered that are like really general in each country?
I think from a scientific perspective, what I think is a misconception is that the vaginal microbiome is considered very simple. So the gut is complex and the vagina is simple because the vagina is less diverse. But we are learning now within our team along the way that this is not the case. It depends how you define diversity. If you go look on species, strain level, if you go look on a functional level, the vagina is much more complex than it seems to be. And another misconception is, and I think Sandra can speak more to that, on how we always thought that it should look a certain way, but it's a Eurocentric point of view. If you only have looked at people from the West or the vaginas of women from a particular country, and then you build your, let's say, health definition on that. But we are seeing in the data of Leke in Cameroon and Laura in Peru that these vaginal microbiomes profiled look much different. Maybe Sandra, you want to add to how that...
Yes, yeah, indeed. This definition of, some people use healthy, others no, yeah, but the dominance of lactobacilli, which is, if crispatus is there, we know then all the scientific evidence support this, but from our results, we see that there are some diverse, bigger, let's say, at least in the Peruvian cohort. That these are the results from the transversal study, but we are checking the cross-sectional study. Now we are running a longitudinal study to really see if this diversity, does it increase or decrease during over 2 menstrual cycles. Yeah, that's from the scientific point of view, and from the societal. I mentioned the antibacterial soaps or the vaginal soaps, which are common, that the women in Peru does is, for example, when they feel a complaint, they feel some uncomfort, it's a vicious circle. They don't feel listened when they go for a consultation with a gynecologist, so they go to buy antibiotics or antifungi without prescription. So there is a lot of use of this antimicrobial creams, and it increases the problem of antimicrobial resistance. So that's also something that is going to improve.
Yes, what I can add is also that women, I think it can be general, because many women think that the vagina is dirty. You have to clean the vagina. Because when I was discussing with women, they say, no, if I don't wash my vagina with soap, if I don't use, because I remember that many women use garlics and aloe vera. I don't know if you know aloe vera, because they have this idea that it's not possible that the vagina can be cleaned alone. So we have to clean the vagina.
Yeah, I guess this is the most extended thing, right? Like using soaps for our vagina. Let's move into the scientific impact. So, okay, now you have all this data from different places. What would be the main factor shaping the vaginal microbiome? Is it the menstrual cycle or the hormones? Are lifestyle more important? Is everything is equally important? What can you tell us about this?
I think this is a question that is also most asked by just local press as well, because everyone is curious. From our data, we see that the largest effects are by age. So the older a woman gets in our data set, we see, because our oldest participant of the first ISALA data set was 98 years old. We saw that then indeed there was a correlation with lesser lactobacilli and more diverse bacteria. We also saw a large effect of women having children. So having had children will have a lasting impact on your vaginal microbiome. But there were different effects. And I think one of the most important things that was important for within our team of studying and the communication was especially communication to non-scientists, was to help them understand the difference between correlation and causation. These were just correlation, not causation. And for us to better understand this is to continue doing follow-up studies. So we were talking about ISALA sisterhood, but within Belgium, we also have this concept called the ISALA daughter projects. And these are selected topics within that we selected together with citizens, and that we continue doing a particular intervention study on to better understand what we see in these correlations. And ending with that is with all of these questionnaires, because the questionnaire took about 45 minutes to fill out, we only could understand 10.4% of the variation. So there's about 90% of a black box of the vaginal microbiome that we still cannot understand. I think there's a lot of other questions that we could have cleaned as well and answered, some stochastic effects, but we're also looking further into maybe genetic immunological effects that are maybe having an impact on the vaginal microbiome. So let's say there is still a lot to uncover. From what we know, we see that age and having birth are one of the largest effects, but sometimes it's also socioeconomic status, but it depends on how diverse your data set is. I would say there's a lot I can say about it, but I would invite everyone to have a look at our paper in Nature Microbiology that was published in 2023. There we go much more in detail.
Okay, yeah, thanks. So yeah, again, could you walk us through some of the key findings so far? Like we have read about these modules, these bacterial gills that you found in the vaginal microbiome. So what do they tell about women's health, what they are, how they are formed?
For our group, it was indeed something, because we're all aware of the community state types, and you have the enterotypes also in the gut microbiome. So we were curious to see what we could find also in this data. However, we are a lab of applied microbiology and biotechnology, and always curious to better understand the underlying biological mechanism. And we didn't want to arbitrarily group particular women. So we first took a glance from a perspective like, are there particular bacteria that are co-occurring more together than with others? And these eventually are being called the modules. Because we see that indeed some bacteria are highly significantly occurring together than they are with others. And we have, in the first paper, we've come to 4 of these modules. So that doesn't mean that one person has one model. You could have all the models in theory. So you'd have like this healthy model with Lactobacillus crispatus, Limosi lactobacillus, and Lactobacillus jenseni. But also pathobionts would correlate together. They're more associated with inflammatory states or BV. One of the most curious ones was a module that had more of gut-related bacteria. So we're really curious to see like what this means. And then it means, and right now in our lab, there are PhD students really working on seeing if these models, why do they co-occur? Do they cross-feed? Do they exchange particular metabolites? Is it strain dependent with synthetic communities? And with these with an anaerobic chamber, with like the bioreactor. So it's very nice that we can go further in not just observing but understanding. I think this is one of the major things that we're continuing on to go on a functional level from the observations that we've made.
Yeah, moving from taxonomy to functions and mechanistic insights and understanding what's out there. Yeah,
indeed.
So these modules that you have just defined it, firstly, I guess, in the ISALA project, were consistent across geography and ethnicity, or do you see different patterns in different populations? Maybe you can tell us about Peru and Cameroon.
Yes, in the Peruvian data set, let's say, our sample size was not as big as ISALA. In ISALA, we have 3,340 women. In Peru, it was 115. And we don't get to have these nice 4 modules, but we see that this co-occurrence between, for example, Lactobacillus crispatus and Limosi lactobacillus reuteri. So we found that one, and also between the pathobionts, just few of them. But yeah, we got there. Yeah, we don't have like a separated module, but we see this similar.
Yeah.
Yeah.
And
Really, Josiane?
yeah.
So concerning the module in the result from Cameroon, actually there is the data analysis ongoing, but the first result that we have now, we see that we have different modes. The result obtained in ISALA is different from the one that we have in Cameroon, because like we we have a module of this lactobacillus, but the lactobacillus is with another anaerobic bacteria, and also we have another module with another anaerobic bacteria, but we are still, as I say, we are still analyzing to try to understand this result, and also to try to figure out all the co-occurrence that we have in our data. And also we have only 108 participants, so we have to take this into account in the data analysis.
Yeah, that makes sense. So yeah, okay. So once you have all this data, no, because you have now very interesting data, and like I guess each woman has its own microbiome. So how do you really translate this information back into the participants? How you do that? I don't know who wants to start.
Well, it's an exercise. I think it's different in every country. So for ISALA, we were first, you do the data analysis and you're curious to see what you find, because at the end of the day, you won't have, not everyone is going to have you have a great profile. So we had to think about that. We did it with our advisory board members and different stakeholders. We presented the results and said like how to balance off. And I think as a scientist, it's very hard to give concise results. We always want to say too much and a lot and nuance. And this is something that we really had to learn to cut back, to keep it simple so that people don't get lost through it all. So we eventually opted for actually quite simple communication. So at first, participants got to know which bacteria was most dominant and how many other people were also part of their, let's say, group. And then you just got a short table, not of all the species. We made a selection of, at the beginning, eight most dominant ones, and now we've expanded it a little bit to 11, I think. And then we had other, and then with a percentage, and then they had a pie chart. Which they really loved. People think it's very sciencey. And each of these bacteria that we selected, we made a passport of it so they could read up on it, on what the bacteria does, where does it occur, what it looks like. And that was actually the first communication. When you didn't have a more favorable profile, we focused a lot on clinical parameters, that people should not panic and they should think if they have particular symptoms. So we let our physicians made a short video listing the symptoms, that if you have them, go to your physician. If not, then this is it. So we try to keep it simple. But it was different in the other
passport concept, yes.
Yes, actually for Peru, we follow the example of ISALA to try to keep it simple. But that simple for the Peruvian reality was still a bit complex. So we had a lot of rounds of discussions with the field workers and other actors to make it simpler. And I remember also some clinicians were saying, why are you going to give the results to the participants? For example, this one that has a Darnerella and Prevotella, which are not good. They will run to the pharmacy to buy antibiotics. And we say, maybe, but we will deliver the results in an information session, and we will make sure that, or to explain clearly that, okay, this is just a snapshot of a moment. The microbiome is dynamic, and you don't have symptoms, because that was the inclusion criteria, no symptoms at the moment of sampling. And yeah, so there was a lot of fear from the clinicians, but in the end, I think the information session was, for them, was clear. It was, we got good feedback, and also we had a long, it was like 4 hours, like we were there discussing. Yeah, it was but it was so, I think that it was so exciting and motivating. So how can I say, you feel like you're contributing, no, to directly to the participants, and they understood. They said it was clear. We also made some videos to explain how to interpret, but yeah, we also made a table, explained the pie chart. Yeah, it went fine. Yeah.
Nice. What about
For
Josiane?
Cameroon, we were inspired by what was done in Peru and also in ISALA. Also, we make the results simple, but for Cameroon, we gave to women the result of the bacterial vaginosis. At that time, we didn't have the result of the vaginal profile, and also we make as simple as possible to try to explain that what it means, because we use the Nugent score to determine the bacterial vaginosis, and we were also have a team, and in the team, because at that time we were also working at the hospital, because I told you that we also enroll pregnant women. So before giving the result to the participant, because we also have some participants with VV positive, we have advice from the medical staff at that time. But I think that as I said, the result was simple. I think that women understand the result, because we didn't have really feedback, just like 2 or 3 women that will contact us to try to understand well what the result means.
Yeah, that makes sense. I mean, I guess with all this project, it comes like a great responsibility, like being able to translate these results in a proper manner to just not chaos, panic, not, So yeah, it sounds challenging also, like for a scientist to know how to do all these things. For that reason, I guess it's very interdisciplinary, your project, right? So yeah, okay. Now, I mean, we have talked mainly about vaginal microbiome, but we know that you go beyond vaginal microbiomes. In fact, recently checking your LinkedIn profile, we have seen this, that you are kind of exploring this potential gut-vaginal axis. So I don't know, we are curious to know the hypothesis behind this.
Yeah, indeed. So like I mentioned earlier, it started with these gut module that we found, which kind of made us think, like, how do these particular bacteria get in the vagina? Is there some reservoir? What's the theory there? And the hypothesis could be whether it is external from the perineum, or it is more of a systemic effect from our just within our bodies, or there's another route. And also from a citizen science perspective, a lot of people were sharing stories and just via social media telling us, like, hey, when I stopped eating sugar, I have less fungal infections. And there are some smaller papers out there linking diabetes and insulin to, for example, fungal infections. So there was something there. So we're curious to study that more in detail, and together also latch on some of our other research questions. So we do that in 2 of our daughter projects, in the Viable study as well, and more in detail in the Rufaida study, where we also focus on the impact of fasting on the gut and the vaginal axis, on the gut and the vaginal microbiome. So we're still analyzing the data there, there's not a lot I can say yet, because we see some things, but you don't want to outcall something while you research a bit more. But ISALA has expanded to just the vaginal microbiome to also, we studied from the beginning already the skin microbiome of intimate body sites, because one of the daughter projects of ISALA studies whether or not you can use the microbiome as an aid in sexual assault tools. And that's our Gene Dope project. And there we also sampled the skin, so more intimate locations. And we also see there, for example, on the breast skin, there's much more lactobacilli than we would have expected on the skin. We always think there's a lot of lactobacilli in the vagina. So there's a lot of questions. As you know, with research, you do a project and you have one question, and with 10, how do these lactobacilli come there? We know from the groin they could have come from the vagina, but all the way to the breast, is it maybe because of a higher hormone level? So there's a lot to explore more, but we're also excited if people have ideas or hypotheses to reach out. And also in our sister projects, they also expand beyond the gut, the vagina, let's say, to other body sites. Curious.
Yeah, that's super interesting. I mean, I don't know if there is any other additional scientific finding that you're willing to share with us, or I don't know, related to vaginal or other microbiomes. There is something.
There's a lot. But
I know.
I think there's hard to choose. I think we try to translate our website now in English, so I would definitely invite people to have a look at the results of these individual daughter studies. So from the Luna project, where we study, for example, the impact of menstrual hygiene products on the vulva and the vaginal microbiome, which has expanded also to Peru and Cameroon. So it's actually going on right now. I think there's a lot of exciting things to expect from that side. So definitely try to follow the group. But also on a species strain level, there's also some interesting findings there, whether mothers and daughters share strains and to which percentage. It's a very technical also, what's a strain, what's sharing. So there's a lot of fundamental research there also. And then on the capacity, the biocapacity of the lactobacille being used in therapeutics, for example. So I think there's that's why we've changed actually ISALA project to ISALA program, because what people kept asking, like, when is it going to end? And now we've come to the conclusion, maybe never. There's just more questions. So we have now the ISALA as a program and different projects within ISALA that they could end.
Yeah, that makes sense. Like, it's an umbrella where a lot of things are happening in parallel. So to all our audience, if you want to know more, just we visit your website and stay connected. Now looking ahead, do you envision translating your findings into tangible health solutions? So are there plans to develop tools or diagnostic tools or products that could directly benefit women's health? So moving from this scientific research that you have, this scientific knowledge, to something that it can be used for women's health?
I think that's our dream. I'm looking at Josiane, that it moves beyond the paper, so that it moves beyond whether it is a diagnostic tool, whether it is to help women, because you can advise them from a preventive point of view, but sometimes you're just unlucky and you're born with having a vaginal microbiome that has more tendencies to be disrupted easily. So we do hope to make it more tangible. There are some people within the group having some entrepreneurial pathways, so we're excited to see them follow through. But I also hope from the sisterhoods that they also have their individual paths as well. And I see Sandra and Josiane nodding. I'm going to let you take over.
Yes, that's something I also want to mention about the sisterhood, that each project has have their own idea, research question, and ambitions. So in some countries it is to develop in the future a probiotic, oral LVP, or in collaboration with us or with other partners. So the idea is that to generate knowledge, but also to translate it to something useful.
Yes, that is true. For example, what I'm doing in Cameroon with the research, I'm not only looking on not only looking on the ecology of the vaginal microbiome, I'm also looking deep on the functionality, with the aim to identify probiotic candidates from our isolated strain.
Yeah, that could be very cool to see some new products for better women's health overall. Okay, so we are moving towards the end. So before we wrap up, could you tell us about this new Umami Research Center of Excellence? What does the creation of this Research Center of Excellence means for the future of ISALA and its global sisterhoods?
Definitely. I think ISALA inspired us that working together and proved over and over again, you get the coolest results and the coolest studies, and bringing a lot of collaborators together. You cannot do it on your own island, with different perspectives. And the Umami Center of Excellence is something that was awarded by international peer reviewers to our group, and it exists of 7 PIs, of which Professor Sarah Lebeer is a lead PI, and or let's say ISALA program fits very nicely within it. Here, the goal is really to advance health equity through a microbiome-centric and interdisciplinary approach, and we focus on women, children, and vulnerable groups. So what we're saying before, this not just with the vaginal microbiome, because the vaginal microbiome is for us the beginning of everything and the most interesting, let's say, niche that we focus on, but it's linked not only to other body sites, but also to if you're giving birth to children. So we're really hoping to expand it, its effects, its relation to other niches, and also to approach it not just from this microbiological perspective, but from other perspectives as well. So this Center of Excellence hopes to do larger studies, work more with international collaborators, and expand even more on our research. So this is also a call to everyone listening to the podcast, that if you want to be involved in one of our project ideas, you can definitely reach out if you want to start up your own sister project. If you have an idea for a consortium that we would love to join and brainstorm together, because we have noticed that with this brainstorm, that as a scientist that you come up with, you can, let's say, tackle these wicked problems better than alone. So that's a bit what Umami stands for, I think. Yeah. I don't know, Sandra, you want to add maybe?
Yes, yeah, and it really fits the role of Umami, or the aims also with this new program we have with Peru. The LEAD IUC program is a research program of 12 years where we will focus on working in the Amazon with a university. It's the National University of the Peruvian Amazon. And yeah, we have last week on Friday was the launch of the UMAMI Center of Excellence, and yeah, we definitely are aiming to work together and to work especially with vulnerable groups, pregnant women, kids, and also remote communities.
Nice. So good to see this, like, professionalizing even more, no, like becoming even a greater, like a larger initiative. So that's great. I don't know if you have any final insights on the expansion of the sisterhood, or how could people get involved, or do you have any exciting new developments in the near future we have not discussed? Anything else that you want to say before we wrap up? Definitely.
Yes, if you're interested in joining the sisterhood, you can always contact me. In the ISALA webpage, we also have information about it if you want to read more. And yes, and the idea of it's that because in ISALA, in the team, we are constantly developing, for example, tools or methodologies, and that's also the aim of the sisterhood, to share this know-how. So yeah, we can definitely help with this, and yeah, we are open to collaborate. Yeah.
Okay, so nice. To close up, I would like to give you a question for all of you. So, like, more about a dreamy question. So what is your biggest wish for the future of women health research, and what challenges do you hope to solve in the coming years? Maybe, I don't know, Josiane, you want to start?
Yes. My big wish is to make women health research more inclusive, because, actually many things have been done up to now. I acknowledge that, but there are still a lot to do. There are still actually many underrepresented groups, and we all know how vaginal microbiome is really important. So it's really pity that we still have women underrepresented group, and also we also have underrepresented topic in the women research.
Nice. I don't know, Sandra?
Yeah, well, for me is that a dream would be like that all the knowledge that we are generating and doing it, that it really reach the women and that they have access, not only women living in urban areas where you have access to everything, but also to women that live in remote communities. And not always also to, for example, that the approach is, hey, let's develop a product and just bring it there, but also valorizing their knowledge, because sometimes they have local knowledge that might work, but it's not scientifically proven, let's say. So for me, it would be ideal to work together and increase the knowledge, and that in the end it favors women's health.
Yeah. What about you,
I think everything has been said. I totally agree with what Josiane and Sandra mentioned. So social impact is really important for research. It's something that's not often emphasized enough. I feel it's a pity. We're very focused on getting enough papers out, but scientific excellence is nothing without really helping and impacting the people. And for me personally, my goal at the beginning of my PhD is I just wanted to be able to help one woman. So if one woman would have come and said like I was helped, that was for me already a success. And that happened also a few times over with ISALA, that women felt helped by having this knowledge, that they were happy with having their results and better understanding themselves. So in a way, let's say I don't want to, in a way for me that was already a little bit reached. And now we are just more ambitious to help more women at all these different aspects. So I would end indeed by saying that the research that we're doing, that it really has the impact that it needs to have. Yeah.
Okay, yeah. Thank you so much for these wishes. We hope they can come true somehow. So yeah, thank you again for this super inspiring conversation. I always really enjoy knowing more about the ISALA project. So yeah, thanks for joining us. And yeah, Josiane, Sandra, Sarah, thank you again for sharing everything with us. Thanks for tuning to the MVIF Conversations, the podcast. Remember to sign him up to our free monthly virtual conference sessions. And until next time, stay curious, stay connected, and don't forget that microbes are everywhere. So thank you.
Thank you for listening to MVIF Conversations, the podcast. Until next time.